dark rings versus light rings is a variation around sort of mean atmospheric pressure. Okay, so this is the amplitude, and then the number of cycles per second is the frequency. Okay, we usually perceive this as loudness. There's some technicalities there in terms of intensity translating to loudness, but you can think of this as the percent of loudness. Okay, so there are three major divisions to the peripheral auditory system, the ear, the outer, middle, and inner ear. So the outer ear is really this, what we usually think of as the ear, which is really just, this sort of cartilaginous flappy opening that we also refer to as a pinna. Okay, and then the middle ear is composed of the three bones that we're going to look at next, and then the inner ear is really, where the action happens. This is the cochlea. These little funny things out here are the semicircular canals that are involved in vestibular function, which we'll talk about in the next lecture. But this coiled up thing right here that looks like a snail is the cochlea, and that's known as the inner ear. Okay, so the bones of the middle ear. So mammals and many other animals are tympanic hearing animals. They have this thing called a tympanic membrane. This is also known as the eardrum. And behind it are a series of three bones. And these three bones, the malleus, incus, and stapes, really function as an impedance matching device. Okay, so there's the pressure in the air, and that has to get translated into pressure within the inner ear, which is a fluid-filled cavity. And so it has to match the impedances between those two. And it also amplifies. And this is probably one of the main reasons that mammals have high frequency hearing, whereas a lot of animals that don't have this sort of three-bone structure are limited to relatively low frequencies. Okay, and the final end of this, the stapes, is connected to what's known as the oval window. Okay, and this is where it really connects into the main part of the cochlea. Okay, and it's the movements of the tympanic membrane that vibrates as those sound pressure waves hit it that transmit the information to the inner ear. Okay, and there's a nice little video of how these bones all kind of work together, but you can look at that on your own. Okay, so now we're going to go and look a little bit at the inner ear. So this is just sort of a more detailed drawing of the pinna, the eustachian tube. For those of you who know, this is something that often gets stopped up with kids. And then here's the cochlea. And connecting the cochlea to the brain is the cochlear nerve, or the spiral ganglion nerve. And this is the eighth nerve. Remember all those cranial nerves? One of them was the eighth nerve, which is the cochlear nerve. And it's bound together with the vestibular nerve that takes information from the semicircular canal. So together these make the eighth. Okay, so just to look a little more closely at the cochlea. So here's that oval window. That's now we've removed the stapes, and that's what's being vibrated during sound. And below it is a little round window. And this this coiled up structure is really a set of three tubes kind of fused together and then coiled up. Okay, and so if you take a cross section through one turn of that tube, you see something like this. Okay, here's one part of the tube. This is called the scala vestibuli, and then this is the scala tympani, and then in the middle you have the scala media. And then sitting sort of in the base of the scala media is what's known as the organ of Corti, and that's where the hair cells are, and that's where the transduction really happens. Okay, so here's a more close-up view of the organ of Corti. Let's back up for a minute. This little purple region here, this is a membrane called the tectorial membrane, and then underneath, sort of forming the base of the organ of Corti, is known as the basilar membrane. Okay, so you have these two membranes that are sandwiching the hair cells in between. And so here they are again. Here's the tectorial membrane, and here's the basilar membrane. Okay, And sitting in between them are these rows of what's known as hair cells. Okay. And then a whole bunch of supporting cells. And there are two basic types of hair cells. There are the inner hair cells. Okay. That's this purple row here. And then there are 3 rows of what's known as outer hair cells. Okay. And in both of these cases, they're sort of the main body of the hair cell. And then you have these stereocilia poking out of the top of the hair cell. Okay. And this is an electron micrograph showing the arrangement of the stereocilia. So each of these little U shapes is an individual hair cell. You can't quite see them as well in the rows of the hair cell, but they're the same. There's this whole series of inner hair cells. But you can see them really clearly in the outer hair cells. It's a really beautiful structure. And we'll talk a little bit more about how this happens. Okay. So let's go back to these 3 tubes. So you have these 3 tubes fused together and then coiled up. So if we uncoil them, they look a little like this. So this is the cochlea uncoiled. So here's the oval window, and then here's the scale of vestibuli, and it's basically a fluid-filled tube that goes all the way down to the end, and then there's an opening, and the fluid goes back around down the skeleton panning and back to the round window. Okay, so it's a closed fluid-filled system. Okay, so you need both the oval window, where you're applying vibrations, and the round window, which is relatively flexible, that allows further vibration, because otherwise you'd have an incompressible system. So you really need to have this system, and what happens is that these waves of pressure flow through this system, and the basilar membrane sits in between. Okay, and the vibrations cause the basilar membrane to vibrate. And it's really kind of a beautiful biomechanical system. There's nothing sort of neural about this particular part of the system. It's just biomechanical. And so it causes a series of a traveling wave of vibration to move along the basilar membrane. Okay. So what does that mean for the hair cells? So the hair cells are sitting here. They're sort of embedded in this base of supporting cells, attached to the basilar membrane. But their stereocilia, the tall one of their stereocilia, is attached to the tectorial membrane. Okay? And so here you have the inner hair cells and the outer hair cells. And there's a real distinction between these inner hair cells and outer hair cells. The inner hair cells are the ones that really carry auditory information to the brain. Those are the primary sensory cells. So they're connected to afferents that send information to the brain. The outer hair cells are not responsible for the main part of auditory information. And we'll talk a little bit more about their function as well. But they're also innervated. And both of these have some degree of afferent and efferent innervation. You guys remember what afferent and efferent are? Afferent carries information to the brain. Efferent carries information from the brain, right? So there's a feedback to the hair cells through this sort of efferent system. Okay, but just remember that the inner hair cells are the primary sensory system for carrying auditory information. Okay, but in both cases, they're attached to this tectorial membrane. So what happens when you have a wave of vibration that flexes the basilar membrane? Okay, so if you have the basilar membrane gets pushed up from its sort of neutral stance, okay, well everything gets kind of pushed up, but because the tectorial membrane is anchored, it provides a shear force on those stereocilia. So, just think of it as something's kind of sandwiched in between, and then when it has to move, there's a bit of a shear there. Okay, and when it goes in the other direction, the shear force is in the other direction. So when you have this vibration go by, you're tipping those stereocilia in one direction or the other. So frequency, I just told you about this wonderful biomechanical system. And it's set up in such a way so that different positions of the basilar membrane are sensitive to different frequencies of vibration. Okay. So frequency is mapped to position along the basilar membrane. And so this is the base end, cochlear base. Okay. So the closest end to the stapes encodes high frequencies best. And as you move along the basilar membrane, it becomes less and less sensitive to high frequencies and more sensitive to low frequencies. Okay. Base versus apex. This is known as the apex. Okay, so what does this look like? So if you apply a vibration of, say, 25 hertz, and you look at the displacement of the basilar membrane, okay, and that's sort of in the y-axis here, versus distance from the stapes. So 0 is next to the stapes, that's the base. 30 is the apex. You'll see that the greatest displacement is at the apex. And then as you increase the sound frequency, so this is like playing a sinusoidal tone, pure tone, the basilar displacement becomes, the peak of the basilar displacement moves leftward toward the base. Okay. And eventually you get much higher frequencies. So we generally hear from about 20 hertz to 20 kilohertz, so 20,000 hertz. So there's a really broad range of hearing. And so at the very base end, you have these higher frequencies. And that's the basis of tuning in the auditory system for frequency. So you can think of it as sort of like a piano keyboard laid out on the basilar membrane. Okay. The other things you can see is that the tuning is not incredibly sharp, right? So there are other positions on either side of the peak that respond somewhat to this frequency. Right? So it's not really super sharp, but there's definitely a mapping of frequency as you go along. Okay, so we're going to I know that's like a lot of information in a very short period of time. So I'm going to play a little video for you that kind of puts it together. Because it's kind of nice to see it. It's a very dynamic system. So it's kind of nice to see it all put together.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch, or frequency, produce a slower rate of vibration. And sounds of lower volume, or amplitude, produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior inguinal ligament are of particular importance for the pivotal axis. Two structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. The stapes moves with a piston-like action, which sends vibrations into a structure called the bony labyrinth. The labyrinth is filled with a fluid called perilymph. If it were a completely closed and inflexible system, the movement of the stapes would be unable to displace the perilymph, and therefore unable to send vibrations into the bony structure. Due to the flexibility of a membrane called the round window, the stapes movement can displace the perilymph, allowing vibrations to enter the labyrinth. The corridor leading to the round window is found within the spiral portion of the bony labyrinth known as the cochlea. Vibrations produced by the stapes are drawn into the spiral system and return to meet the round window. The portion of the spiral passage in which vibrations ascend to the apex of the cochlea is called the scala vestibuli. The descending portion of the passage is called the scala tympani. A third structure, called the cochlear duct, is situated between the scala vestibuli and the scala tympani. The cochlear duct is filled with a fluid called endolymph, and when viewed in cross-section, the membranes separating the two fluid-filled systems are visible. They are Reissner's membrane and the basilar membrane. The membranes are flexible and move in response to the vibrations traveling up the scala vestibuli. The movements of the membranes then send vibrations back down to the scala tympani. A specialized structure called the organ of Corti is situated on the basilar membrane. As the basilar membrane vibrates, the organ of Corti is stimulated, which sends nerve impulses to the brain via the cochlear nerve. The actual nerve impulses are generated by specialized cells within the organ of Corti called hair cells. The hair cells are closely covered by a structure called the tectorial membrane. As the basilar membrane vibrates, the tiny clusters of hairs are bent against the tectorial membrane, triggering the hair cells to fire. The entire basilar membrane does not vibrate simultaneously. Instead, specific areas along the basilar membrane move variably in response to different frequencies of sound. Lower frequencies vibrate the basilar membrane closer to the apex of the cochlea, whereas higher frequencies produce vibrations closer to the base. This arrangement is known as tonotopic organization. Together, this sequence of events is responsible for our acoustic perception of the world around us.
very nice demonstration of sort of how sound travels through this system in a way I could not do with any kind of hand waving. So there we go. So let's move on to, didn't get into exactly how do those hair cells transduce those vibrations into nerve impulses. That's really what we're getting at. Okay. So if we look closely at those hair cells, here's a electron micrograph of a set of hair cells, and this is sort of this sort of stereocilia bundle here of the inner hair cell, and there are actually 3 rows of hair cells here. So 1, 2, 3, and they're connected. And if you look very closely, they're connected by this little string which is called a tip link. Okay. And so the system is literally a mechanosensitive system with a little string. So when these are when the stereocilia are displaced, it pulls on the little string, which opens a mechanosensitive channel. Which seems really crude, but that's what it is. It's literally pulling a channel on a string. Okay, and now we know a little bit more about some of the proteins involved in this. That this is a type of cadherin that connects 2 channels. So this is one of those trip channels again. Remember we talked about the whole family of trip channels being mechanosensitive. So this is a particular one, trip A1. And then there's a whole lot of actin. And so there's a lot of interesting protein interactions here that we don't really have time to get into. But those are sort of the main ones. So it's literally a mechanosensitive system. Okay, so at rest, or in sort of the hyperpolarizing direction, these channels are closed, and the tip links are relatively slack. And then when you bend the stereocilia, the tip links pull on the channels and open them, the trip A channels. It's a cation channel. And you have potassium ions that flow inward and cause depolarization. The depolarization causes calcium influx, which causes neurotransmitter release from vesicles onto the auditory nerve afferents that are sitting here at the base waiting to be activated. All right. So the depolarization is because of potassium flowing into the stereocilia. That strike anybody as odd? Yeah, at least one person? What's different? The potassium is flowing. Potassium's flowing in. What did we usually say? Potassium flowing out. Okay, so weird. Why is this happening? Okay, we've always talked about potassium as being hyperpolarizing, not depolarizing, because it's flowing out. Okay, so what does this tell you about how the concentrations of potassium are?
So outside is more than inside.
That outside is more than inside. Okay, that's a little unusual. Okay, well it turns out that this is a highly specialized system in which the extracellular media bathing the top half, these stereocilia, that are sort of poking above this little line here, we'll call it, is bathed in a high potassium endolymph. Remember that the middle of the scale media contained a fluid that they called endolymph. And it turns out to have very high levels of potassium. Okay. And the bottom half of the cell is in what we would normally consider to be the type of media that we talked about all other neurons. Okay. And this works similarly both for hair cells in the auditory and the vestibular system. So let's look at this a little closer. So here's sort of another cartoon. We have our stereocilia, and here's the main body of the hair cell. And you have all of these, remember all those supporting cells that those hair cells are sitting in the middle? Well, it turns out that there's a really tight junction between the top of the hair cell and the supporting cell. And that separates this endolymph fluid from the extracellular medium bathing the bottom. Okay, so here's your scale media. This is the endolymph that has high potassium. In the rest of these systems, the skeleton panning is known as perilymph, but importantly, that this is where you have the low potassium. relative to the inside of the hair cell. Okay. And so in the bottom, you have the potential to have potassium leaving. Okay, so potassium can flow in the stereocilia, but flow out the main body of the cell. It's a little reminiscent of the photoreceptors, right? Where you have sort of flowing in and then flowing out. Okay. And the reason you can have this is because you have this really tight seal between the two areas, between the endolymph and the perilymph. Okay. It's kind of a cool system. Okay. And the potentials reflect this. So if we consider the outside here to be 0, that the inside of the hair cell is something like -45, perilymph 0, -45. The endolymph is very positive, just like you would expect based on the potassium concentrations. All right, so when you have deflection of that stereocilia, you have an influx of potassium ions which depolarize, and it turns out that this can happen really rapidly, okay, relatively rapidly. So when you have some hair cells, so these are all recordings of hair cells. This is membrane potential from hair cells that are tuned to different frequencies. So this is tuned to 300 hertz, 500 hertz, and so on, 1,000 hertz, 3,000 hertz, and so on. And so if the frequency is relatively low, so those vibrations back and forth is low, and the stereocilia are vibrating back and forth relatively low, the membrane potential can actually follow that. Okay, and so each of these deflections is a back and forth vibration of the stereocilia. Okay. But as the vibrations get a higher frequency, the hair cells can't follow it at that frequency, and you end up with basically a smooth depolarizing plateau here. And the reason that you don't just average everything out is because there's a bias toward depolarization. So when the hair bundle is displaced in the depolarizing direction, you get a fairly large depolarization. In the opposite direction, you have relatively little change in potential. So it's biased toward the depolarizing end. And that's why you get this sort of plateau potential. So it's interesting because it means that at relatively low frequencies, you can use that oscillation of the hair cell as an indicator of what frequency is being played, or what frequency it's responding to. Okay. And at higher frequencies, the hair cells that are sort of sitting on that, the higher frequency end of the basal membrane, all they can tell you is that they've been activated. Okay. So you actually have sort of two systems of encoding frequency. One is you have the frequency explicitly in the oscillations of the membrane potential. And the other is you have which of these hair cells are active. So if you have a 3,000 hertz, a 3 kilohertz sound, this hair cell is going to be active, this one's going to be relatively inactive. So in this sense, you have a positional map of frequency, which hair cells are active. Okay, so pitch at high frequencies can be determined from the position of the stimulated hair cell and the afferent that's innervating that hair cell along the basal membrane. And so in that sense, you have this tonotopic map. It's a map of position along the basal membrane because the basal membrane is tuned to frequency. Okay, so let's start looking at the auditory nerve. So if you record from a single auditory nerve, each nerve innervates essentially a single hair cell. Okay, so it's going to be responsive to whatever that hair cell is going to be responsive. And you're going to get tuning that looks like this. So let me kind of explain what these are. So these are 6 individual auditory nerve fibers. So if you record from one individual auditory nerve fiber, what you typically do is you play a pure tone at a certain sound level, and then you increase the level, you go from soft to loud until you start to see a response in your neuron. Okay. And then you mark what that sound level is. So we have sound level on the y-axis and frequency on the x-axis. Okay. And so the softer the sound is, that activates the nerve fiber, the lower this number. So this is known as a threshold plot. So this tells you what amplitude is the threshold amplitude for that frequency, for that neuron. Okay, and so the negative peak here is the best frequency for that axon. That make sense to everybody? You're finding the threshold amplitude for each neuron. And so each of these has a peak at a different position along the frequency axis. And we can kind of line those up. So if we line them up, that looks here. Just want to make one note. I'm not crazy about the way that they've labeled the intensity here. So this is in decibels, but for some reason they've labeled it in decibels relative to some loud reference. So the bottom of this peak is really about, if you look at actual auditory nerve papers, is actually about the level of human hearing. So 0 dB SPL is what's considered the audibility threshold for human hearing. So 0 dB sound pressure level. Okay, so most everything that you see is like relative to what humans can hear. And so the baseline down here is human hearing. Okay, so you can plot each of these. And so each of these fibers, we can say, is due to innervation at a particular position along the cochlea. So now we sort of flipped it. The base is over here, and the apex is over here, right? Yeah, relatively low frequencies, relatively high frequencies. So again, that's that tonotopy, that mapping of frequency on the inner ear. Let me back up just for a minute. So we said that that the hair cell can follow the oscillation of the sound vibration. Well, the nerves can also follow the vibration of the sound frequency as well. And this is known as phase locking. Okay? And this is a very important component of processing in the auditory system. The fact that the auditory nerve can follow those vibrations up to a certain level. Say below about 2 or 3 kilohertz for humans, using the phase lock. Okay. And so what that looks like, if you record from a nerve over many repetitions and create a histogram of when it spikes, okay, this is the sound stimulus, you can find that you get this very peaked histogram. So it tends to spike at a particular point along the sine wave of the pure tone. And that's the phase part of the phase locking. So it's locking onto a particular phase of the sound stimulus. Okay, so again, in theory, you could just sort of count the interval between these spikes and take a really good guess as to what the frequency was that you used as a stimulus. Okay. But that only works up to a certain number, certain types of frequencies. The other reason that phase locking is really critical is, as we'll see later in the lecture, that we can use phase, or the timing of these spikes relative to the sound, as a cue for comparing across 2 different ears for the purpose of sound localization. Okay, so let's return for the moment to those outer hair cells, which really attract a lot of attention. People like to study these because they're really so beautiful. They're all nicely lined up. They've got this little curve to them. There's 3 of them. They're really beautiful. Okay, but what do they do? And you know, I think we're still kind of working on this aspect, even though it's been really, really well studied. And a lot of what we know about hair cells is derived from study of the outer hair cell as opposed to the inner hair cell. They're just a little more accessible. Okay. And interestingly, the outer hair cells move. Okay. A few slides ago, you probably noticed a lot of actin and myosin. And so it turns out that these cells are fairly motile. Okay. When they are stimulated by vibrations, they'll move in response to the vibration. Okay, so it's not, these hair cells are just sort of passively listening to these vibrations, they're actually, by virtue of the fact that they're connected to the pectoral membrane, are modifying the system at the same time. And it turns out that their movement is such that they provide an active feedback that amplifies the movement of the basilar membrane. And so they actually help sharpen the tuning of the basilar membrane to particular sound frequencies. It's a very cool system, but it's still, we're still trying to understand why it's happening. In addition, those efferent fibers that we talked about that contact the outer hair cells can actually modify the vibration of the basilar membrane, and it's thought that this also helps by, on the one hand it can amplify sound, but on the other hand it can dampen the sound response. So you have a whole system that's set up to kind of respond appropriately to sound levels. Okay. And then finally, the movement of these hair cells can actually cause sound coming out of the ear. Okay, so the system is set up so that it can actually cause vibrations that you can hear. If you put a little teeny tiny microphone in your ear, you can hear these vibrations. These are called otoacoustic emissions, OAE, and they're actually used in newborns to test for hearing. Okay, so anybody who's had a new baby, one of the first things they come in, besides they give them vitamin K, they stick them and get their blood. But one of the things that they do is they check their hearing, the very first couple of days, and it's these otoacoustic emissions that are the signal that determine that baby has good hearing. All right, so just for fun, we're going to look at this video.
One, two, three.
Okay, before we get started, you're going to hear music. We're not actually playing music to the hair cell. Okay, it's not in an ear, can't hear. What's happening is that there's, oh, you can't see this. Where'd it go? So what's happening is that there's an electrode, and the electrical signal via the electrode is being played into this isolated hair cell. So this is an isolated hair cell sitting in a dish. Okay, and the only stimulus it's getting is
8 o'clock, 4 o'clock, rock.
Electrode.
5, 6, 7 o'clock, 8 o'clock, rock. 9, 10, 11 o'clock, 12 o'clock, rock. We're gonna rock around 2 o'clock tonight. Put your glad rags on. Join me home. Have some fun when the clock strikes 1. Rock around the clock tonight. Rock, 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 rock. Rock it out. Rock around the clock tonight.
all right. You get the picture. Okay, so it's moving. It's literally moving to the signal. Just in case you weren't sure. Okay, so hair cells, inner hair cells, outer hair cells, the peripheral system, incredibly acute. It can hear over an enormous range of sounds, but it's also very sensitive. Okay. And so one of the major things that we want to talk about today is this idea of hearing loss. So it's incredibly common to have hearing loss, and there are a number of different phenomenologies that are going on here. And a lot of it has to do with the fact that those hair cells are quite sensitive. Those little stereocilia sitting out there are very sensitive to damage. Okay, so a number of different ways that you can damage the ear. It's a very long list, but we'll just cover a few of them. Otitis media is the middle ear infection that kids often have. So if you have prolonged or acute ear infections, it can cause long-lasting hearing damage, either by rupturing the tympanum or other aspects to the middle ear there. Okay, so that's very, very common. There are a number of what we call ototoxic drugs. So these are drugs that can destroy the hair cells. They're quite sensitive to a number of different agents, among them antibiotics, particular kinds of antibiotics, and chemotherapy agents. And so you can damage the inner and outer hair cells that way. There are a number of genetic factors. There are a number of interesting family histories of HL is for hearing loss. One in particular is this connexin 26 mutation. Anybody remember what connexins are? Back in our synapse lectures. Connexin, it's one of the gap junction proteins. Okay, and it's still not really understood how this works. It's thought that the connexin is maybe in the supporting cells, and so connecting a number of the supporting cells, and so a mutation in this gene can cause a hereditary form of hearing loss. Okay, and probably one of the most prevalent things is noise damage. So, you know, you've been told from a very young age, don't play those earbuds too loud, turn down the music. Okay, and so there is chronic cumulative effects of playing very loud sounds. Okay. Sounds that are not loud enough to, you know, burst your eardrum, but over a very long period of time, it accumulates and causes damage. Yeah.
How do antibiotics hurt hair cells?
So how do antibiotics hurt hair cells? I think the mechanism is because the hair cells have a lot of cytoskeletal proteins, and those are somehow sensitive to particular kinds of drugs. Yeah. I don't know the exact mechanism, but I think it has something to do with that. Yeah. Yeah. Okay. And then, you know, you're probably all quite aware that if you have very acute loud sounds, and then 90 to 100 decibels, that you can have, you know, immediate and long-lasting hearing damage. Okay. A lot of this has to do, you know, even if the tympanum is not damaged, that you can damage those hair cells. And the more you damage the hair cells, the more you have hearing loss. Okay. And in particular, if you are exposed to very loud blasts, like an explosion or gunshots, and so on. Okay. And then presbycusis is the term for age-related hearing loss. So we all know that our hearing gets worse as we get older. So it has a particular name, and it's displayed as elevated thresholds. So sounds have to be louder to be detectable. This tends to affect your high-frequency hearing before your low-frequency hearing. So that's why old people like me can't hear the higher frequency sounds so well anymore. But one thing about this is that it's not entirely clear what component is strictly age and what part of it is just that cumulative noise damage. Because we're all exposed to noise constantly over our lives. People are still trying to kind of sort that out. Okay, there's another condition that's known as sort of generically as auditory neuropathy. And this is a case where, you know, the thresholds are high. You go to an audiologist and they say your hearing's fine, and you're like, but I can't hear very well. You know, I can't hear conversations in a noisy restaurant, or I can't hear what they're saying on the television. And so this is a form of pathology known as auditory neuropathy. And people still don't really understand what's happening here. One possibility is that those phase locking signals are getting lost along the way. They're not being processed properly, and you really need those in order to differentiate signal and noise. And then finally, there's a pathology called tinnitus or tinnitus. This is that ringing of the ear. It's very, very common, but it's especially common when you've had peripheral hearing loss. Okay, so you've had noise damage, or you had damage due to chemotherapy drugs, and you start to hear this ringing in your ears. very annoying, and it ranges anywhere from just sort of mildly annoying to actually debilitating. And the idea is that you've had some sort of loss at the periphery, and then your central structures are kind of trying to overcome that. They're trying to compensate for that loss of input. And this is really the #1 disability among veterans. So veterans are very, there's a very high prevalence of tinnitus among veterans. And so the DoD is very interested in coming up with effective therapies, and right now there really isn't a lot to do for tinnitus. Okay. So one last aspect of hearing loss I wanted to talk about is cochlear implants. So among roughly about 100,000 people, Americans, now have cochlear implants, either in one ear, some of them are in 2 ears. These are people who have profound hearing loss, not just mildly hard of hearing, but profound hearing loss. And in this case, they can basically get this prosthesis. I mean, this is really sort of, the future of neuroprosthesis. And now it's sort of normal to everybody, right? So you've probably all know somebody or met somebody who has a cochlear implant. And the idea is that you can, let's start with the electrode. So basically, the idea is that you've lost your hair cells at the periphery, but you still have an intact auditory nerve, okay? And so you can insert an electrode. So it's an electrode array, so it's a whole set of electrodes along, you sort of snake them into the cochlea, and then you have a microphone on the outside and a processor, a preprocessor, that will then electrically stimulate the auditory nerve. And so low sounds can be stimulated on one end and high sounds on the other. And so you can translate the sound in the environment into electrical stimulation of the auditory nerve. Yes?
Since these devices are usually reserved for younger children, are there any long-term complications or negative interactions with the inner ear after inserting these?
So one of the interesting things is that it's not limited to young children. Young children do best in learning how to interpret speech quickly. And so the earlier people get implanted, the better they do. But actually older people are implanted. I have an 80-something-year-old aunt that now has a cochlear implant. She lost her hearing in her 60s or thereabouts.
Yeah, but I guess the question was more about since I guess orthotics have a tendency to degrade over time. It seems like something like a device that was implanted so early in a childhood would likely degrade. Is there toxicity that comes from that?
Not that I know of. I mean, in that sense, it's been very successful. It's not like the electrode array has been rejected.
You can just Google
in
that
some
shit
cases,
for
you can
instance.
have scar tissue that can impede the electrode stimulating the auditory nerve. I can think of there's a couple of groups on campus that work with cochlear implant patients, and I can think of one. We're talking about one example of somebody who had their implant taken out and then reimplanted. You can have issues because over age you will lose nerve fibers, and so it may become less effective. But as far as I know, there's not a lot of negative consequences to long-term implantation.
And does these inner ear, does the cochlea grow as you age? So you need multiple cochlear implants throughout?
That's a really good question. I don't know. I don't know the answer to that question. Yeah, I'd be guessing. Yeah, I don't know. But that would be a good question. If it grew too much, then you'd have to take out one implant and put another, and then you'd risk having scar tissue, those sorts of things. As far as I've read, that's not a big issue. But that's a good question. That's something that you have to think of. This is one area where you may be able to get some really good longitudinal studies on that aspect. Yeah. Okay, so, but one of the things that really interests me about cochlear implants is that initially when it's implanted, people just sort of hear noise. And you can implant these in people who, have never had hearing or have never had hearing before they've understood or spoken themselves in the case of children. And they can learn. And the positioning of this electrode, the electrode, you can only get it so far in. So the positioning is actually a little bit mismatched with the actual frequencies that part of the cochlea is supposed to be hearing. And people learn. So there's some sort of plasticity in the central nervous system that allows people to make sense of these signals. And in that sense, it's a very crude system, and yet people learn to understand speech quite well.
And that's not age dependent?
It is a little bit age dependent. The younger they are when they're implanted, it seems like the more plastic they are, the better they can sort of overcome. Older people may have less plasticity, like adults may have less plasticity. Yeah, so there's definitely an age dependence in how quickly they get up to sort of an acceptable level. All right, so let's move on. We may or may not finish all of this today, but let's move on. So I want to talk about sort of the next level. So the auditory nerve goes on into the brainstem. And either today or tomorrow we'll talk about localization and clinical processing. So let's start to talk about these central pathways. And really the periphery has been really well studied, and there's a lot yet to do, I think, in the central area. And it hasn't really been as well studied as the visual system, especially at the cortical level. I mean, there's a lot of work yet to do. Okay, so cochlea. You have the auditory nerve fibers that then project to the first level in the brainstem, which is called the cochlear nucleus. Okay, so that receives auditory nerve input. And then, roughly speaking, the cochlear nucleus sends information to the next level, which is called the superior olivary nuclei, which is located in the pons. And then there are some lemniscal nuclei that do some processing. And then finally you have the inferior colliculus in the midbrain. There are also direct projections from cochlear nucleus to inferior colliculus. But really all auditory information has to go through the inferior colliculus before it goes on to thalamus and then to the primary auditory cortex called A1. Okay. And this is a hugely simplified version of these ascending pathways. You have both from the cochlear nucleus onward, you have both bilateral and contralateral ascending pathways. But one of the common themes of auditory processing in the system is that you have multiple parallel pathways that are involved in encoding different aspects of sound. And we're not going to get into a lot of detail on that. We'll hit on sort of two particular examples of that. Okay, so this is sort of a simplified box plot. Now I'm going to show you briefly the sort of the typical textbook version of this. So here's the cochlea projecting to the cochlear nuclei, and there are actually 3 divisions of the cochlear nuclei. We don't really need to get into what's what for now. Then there's bilateral projections to the superior olive. Here's the lateral meniscus. Here's the inferior colliculus, and then you have thalamus and finally cortex. The auditory cortex is located in the temporal lobe of the cortex. Okay, and here's basically the same thing again in words. Auditory nerve to cochlear nucleus to superior olive. Inferior colliculus acquired, projects to in the thalamus. The particular nucleus is the medial geniculate nucleus. Remember, vision is the lateral geniculate nucleus. So you have those 2 systems in the thalamus. Okay, and then they send to the auditory cortex. Okay, and sort of 2 of the main themes that I want you to remember from from this general system is the idea that you have these parallel organizations, that at each of these levels, information is sort of split into different streams, processed, and then comes back together again. And I'll show you an example of that. But also that at each level of the system, there is tonotopic. So remember, frequency along the basal membrane. Tonotopic mapping is at every level along the system. So frequency is sort of one of the dominating factors in processing the system. Okay. All right. So probably the best studied example of auditory processing is the processing of sound localization. So really critical information as an animal out in the world is where is that sound coming from? Either you're, a little bunny prey and you want to know which direction that predator is coming at you, or you're a predator and you want to know where your prey is. So sound localization. So there are two major divisions of the superior olivary complex, the medial and the lateral superior olivary, abbreviated MSO and LSO. And these are the two major sound brain areas where you can compute two cues for sound localization. Okay, and we'll get on to exactly which cues we're talking about. But the idea is that they get information from the left and the right side. Okay, and this is known as binaural information, two ears, oral, anything that has oral is sort of auditory. So binaural, analogous to binocular for vision. Okay, and one of the important things to remember is that location in space is not explicitly represented in the ear. All right, we talked about the eye having a retinotopic mapping. So position on the eye correlates to a position in space. Position next to that position correlates to a position next to the position on the eye. So you literally have a map of space on your eye. And likewise with somatosensation, you literally have a map of body surface into the brain. Okay, position. Okay, auditory information, that information is just not there, right? Not there on the position of the basal membrane. You have to compute it. And that's what makes this kind of an interesting system to a lot of us. You have to compute localization in space. So how do you do that? Okay, and so there are two major cues that you can use. One is called the interoral time difference, and that's processed in the MSO. And the other is called the interoral intensity difference. So remember, oral, interoral, between the ears, time differences and intensity differences. Okay, so what do I mean by those? Okay, so you remember that sound is a sinusoidal pressure wave. Okay, so when a sound arises on one side of your head, okay, the pressure wave has to arrive at one ear a little sooner than it arrives at the other ear. So there's a difference. The size of your head determines how much faster it arrives at one ear than the other. And that's the interoral time difference. Okay. And likewise, your head's kind of a bit of a shadowing object. And so sound on one side is louder in one ear than the other. And so that's what we mean by interoral time differences is, how many wavelengths more does it take to get to one ear than the other. And interoral intensity differences, so we abbreviate ILD, L is for level, it's just easier to say than IID. Level difference is because of the sound shadow. And obviously the bigger your head, the larger that cue is. So small animals actually have a really hard time localizing sound. And bigger animals like us can use sound localization cues like this. Okay, so because we have symmetrical ears on either side of our head, both of these cues are horizontal cues, right, left to right. As you go up and down, you don't really get any information from these cues because it's the same to either ear as you go up and down. There's an animal that actually uses both of these cues to find two-dimensional space. And this is the barn owl. And if you end up taking the 400 level classes in neurophys or whatever, you'll learn more about the system. But the basic idea is that barn owls are incredible auditory predators, and they can localize cues in complete darkness. So we know that they have great auditory localization abilities. And they can use these two cues. They're set up so that they'll use timing cues for the horizontal localization, but they'll use level difference cues in the vertical dimension. And this is because they have ears. You can't see them because of all the feathers, but they have ears where one is sort of canted up and the other's canted down. So they're asymmetrical. So something above is louder in one ear than the other. Something below is louder in the other ear than the one. Okay, so it basically has converted one of these cues into a vertical cue. Yes?
You said determined by the number of wavelengths between like the time at which the ears affect the noise.
Kind of, yeah.
So is that why, or would that imply that we are worse at locating very low frequency sounds?
So the timing cues rely heavily on being able to encode the phase of the sinusoid, which we can only do at low frequencies. So we only have information about the timing cues at relatively low frequencies. The head shadowing effects, on the other hand, are better at higher frequencies than at lower frequencies, which is kind of a wavelength effect. So we tend to use high frequencies or ILD cues. So I wasn't going to kind of get into that, but there's this theory that we use low frequencies for timing and high frequencies for intensity. There's a little overlap in between. So barn owls, it turns out, are incredibly good at phase locking at higher frequencies than we are. So they have an extended region in which they can use these timing differences. Okay, and I'll just point out that here's an example of an auditory stimulus wave arriving at the left ear, say a little bit sooner than at the right ear, and the amplitude of this stimulus is a little bigger in the left ear than in the right ear. Okay, and so it can use both of those cues to localize in space. And so it turns out that the barn owl can combine those two cues at the level of the inferior colliculus to create a map of space. Okay, and this has been shown. Okay, so here's a barn owl sort of sitting in a three-dimensional world, and this is the inferior colliculus, and so it has a map of auditory space. So 0 is directly in front of it, and then you go from 10 degrees in one direction to 40 degrees in the other direction. Is in this direction. It's not showing the elevation, but basically you have a 3D map, or sort of 2D map of space in the inferior colliculus. Okay, so how do you create these cues? Okay. So the medial superior olive, the MSO, has another name in the barn owl, but we won't bother with that, is set up in a way to compare information from these two different ears. And it's set up in a way so that it compares the timing of the information in the two different ears. Remember we said that the auditory nerve inputs are phase locked? Okay, so if the sinusoid is shifted in phase, the firing is also shifted in phase. Let's see if I can draw that.
Is this for the barn owl or for?
Let me come back to that. Okay. So the idea is that in the left ear, you have some sinusoid. And you have firing at a particular phase coming from the left ear. In the right ear, this is absolute 0 time. In the right ear, so let's say that you have a shift in the phase. do this properly. Like that. And if you have sort of a similar phase locking of the firing, okay, so now you have a shift in the sine wave is translated into a shift in the firing, in the two nerves input. And let's just talk about this as if we're talking about nerves. There's actually a relay step. Don't forget about that. The phase information is maintained in that relay step. And then in the MSO, you have an array of neurons. And that's represented in this drawing as A, B, C, D, E. Okay? And the idea is that these neurons each act as coincidence detectors. So they fire maximally when they get inputs from either side that arrive simultaneously. Okay? Not very much otherwise. So if you have a sound, say from the left side, it's going to arrive at the left ear, okay, first, and then it's going to send an impulse along these axons into the MSO. Okay? And these axons form what we call a delay line. Okay, remember we talked about conduction velocity? So there's a finite velocity along those axons. So the longer the axon, the longer it takes for the action potential to go down the axon. Okay, so it goes down the axon and arrives at neuron E. In the meantime, sound has arrived at the right ear, fires off an action potential, and in a very short time arrives at neuron E. So if the sound is off to one side, it takes a longer path to get to E and a shorter path to get to E from each ear, and arriving simultaneously here. Okay, and so E is going to fire. If the sound were from directly in front, you would need an equal path to get to C, and now C would respond. So each of these neurons is going to be sensitive to a particular position in space. Okay? And so it's the combination of having coincidence detector and having these delay lines that are compensating for the delay in the arrival of the ear. Does that make sense to everybody? So it's basically a map of space in the MSO. Okay, slightly, wait, go ahead.
So the longer axon is compensating for how longer it takes to get to other. Okay.
The longer axon is compensating for the delay in the arrival at each ear, and vice versa.
Thank you.
Okay, a slightly less complicated system is set up to detect level differences. Okay, because you don't have to worry about phasic timing, the extreme timing. And the basic idea is that you have inputs to the lateral superior olive. So you have a direct excitatory input, and then the opposite side sends an input to this nucleus, we're just going to call MMTB, which is inhibitory. So there's an inhibitory relay. And so you have excitation ipsilateral, inhibition contralateral, and it's the balance between those two that determines whether sounds are louder on one side or louder on the other side. So if it's equally loud, then they cancel out. If it's louder on one side than on the other, the excitation is going to win because the excitation will be stronger than the inhibition from the opposite side. And so you get a tuning that looks like this. That something that is louder on the left excites the left LSO, something that's louder on the right excites the right LSO. And then you can further up in the system, you compare the left and the right, and you determine exactly where that signal is coming from. Or in the case of the barn owl, where it's coming from in the foundation. All right, so why don't we stop there? Let me ask you, does anybody have any questions about what we covered today? Yes?
Can you just go back and explain the ABCD stuff again?
Sure.
Because like what I I don't understand is like why is E the leading neuron when it has the longer axon?
Right. So what you would do is you would add up the time. So the question was, why is E the leading neuron when the delay is longer? So what you want to do is you want to add up the time from the loudspeaker to the ear, which is short here, and the delay from the ear to that neuron, which is long.
Oh, okay.
In the right ear, you have a long delay to the ear and a short path to E. So you want the summation of those two to be equal.
Is there any information about the distribution of hair cells throughout the cochlear path?
Sorry?
Is there any information about the distribution of hair cells throughout the cochlear path that you're crossing?
Well, so the hair cells